Hi, my name is Jerome Staal. I'm a senior editor at Nature Medicine, and today I'm at the 87th Cold Spring Harbor Laboratory Symposium on stem cells, and delighted to be joined by Professor Tuveson and to discuss some of the work that he's doing and also a bit about his talk today. Delighted to have you.
Thank you. Thanks a lot, Jerome. Please call me Dave. I'm a professor at Cold Spring Harbor, and I am honored to be invited to participate in an annual symposia here. I participated in one in 2005, which was on cancer. And so this symposia on stem cells is actually very interesting to me because I would say over the span of my career, I could see how the field of developmental biology has become an extremely important aspect for how we now think of cancer. And I've learned a lot in the day that I've been watching the lectures, and I'm hoping to talk about a few topics when I give my lecture on how the field of stem cells has really opened things up for many of us in the cancer field.
As a bit of background, could you give us an idea of the type of research that's been done in your lab?
Sure. So my laboratory works on pancreas cancer, specifically the ductal form of pancreatic cancer. And this is a bad disease where most patients who get pancreas cancer don't survive the diagnosis. When I started working on it 25 years ago, it was a mysterious disease, and now it's not mysterious, it's just still terrible. We know a lot about this disease because we have been lucky enough to create model systems for this cancer. First in mice using genetic engineering, similar to the approaches that developmental biologists first pioneered: embryonic stem cells, altering genes, injecting them in blastocysts, creating mice, breeding mice together so that the genes of relevance in ductal pancreas cancer are expressed in the pancreas of a mouse. And then, being fortunate that in fact that's what happened, and early cancers give rise to advanced cancers, which then spread through the animal. And so the model system really allowed us to ask questions that we could never in humans. Mm-hmm. And the fascinating thing, again, is that almost every step of the process that we've described involves some aspect of developmental biology and how when it works well, there's health, and when it just is subtly changed, there can be disease, like cancer. Mm-hmm. And so these mouse models of pancreas cancer have been a finding that has allowed a field to be nucleated. And when I started working on pancreas cancer, there were probably 10 laboratories in the world who worked on pancreas cancer, and now we have hundreds. And that's great, but it also means we should have better health benefits for our patients. That's been hard, but we have these mouse models, and more recently, which I think is how I won this invitation for this conference, is we got inspired by Hans Clevers when our lab was in Cambridge, UK, about these new tissue-based models that he had derived ex vivo, which he termed organoids. And I was really impressed by the progress he was making in the intestine, and I teased him, it would be great to do this in the pancreas, and he teased me back, only if you help. And these are just snippets of a conversation, but it led to a really meaningful collaboration, and we were really delighted to have that opportunity to work with him and his team to develop pancreas organoids, and then finally pancreas cancer organoids from humans, as well as from various animal species. And this is now a system that allows us to look at a disease where we could never do from patient samples or even samples from the mice, because you can only ask so many questions in a pure in vivo setting. You need to reconstitute in vitro to allow you to ask, how does step A give rise to B to C to D? And many of these steps are developmental biology pathways. Cell communication between epithelial cells and mesenchymal cells, for example, and recognition by the immune system, all of the things that we now think of. And so it's really great. And when I was taught about developmental biology, some time ago as a undergraduate, medical graduate student, et cetera, the field was nascent. It was, as I said earlier, descriptive. It was, we see these processes and we can give names to them, but there was no clear understanding of things. Pioneering genetics in Drosophila and in worms led to the identification of mutants with, entertaining names, which essentially identified pathways, many of which are our developmental pathways, the 4 or 5 fundamental developmental pathways. And going from observational to molecular and developmental biology really was in parallel for cancer, where we were looking for cancer genes, which were what we call causative in the process. And I think merging the developmental biology knowledge with the cancer genetics knowledge has led us to where we are now, where we can study this disease. And we still have work to do, but it is so rich and exciting.
So you have these fantastic tools, these tissue-based tools. You got the mouse models. You have some patient data. What are the big questions right now in the field? What are the big questions that are being asked of these models, basically?
I think the big questions being asked of the model are they span the gamut from fundamental biology all the way to clinical application. And on the fundamental side, the questions we have are very similar to the embryologists. What are the different cell types that you need to put together to actually elicit a response in a cell that carries mutations such that it now acts like a cell with malignant potential? We now know, even though Bob Weinberg has a book where it says, one renegade cell, it's actually one renegade, a pack of cells. There's not one renegade cell that's going to do anything wrong. It's actually a group of cells which cause problems. And yes, you need a cell with mutations, no question about it, but you need cells around it that cooperate with it, that support it, that elicit a malignant phenotype. And so a fundamental question is, what are the other cell types that surrounding a cell with malignant genetic signatures will elicit a malignant behavior? And so, in our pre-interview conversation, I talked about we all have all kinds of malignant cells in our body. They just don't look that way. They don't act that way. We carry oncogenomes everywhere. And that shouldn't depress people. It should make you curious. Why? Why is it that the cells, are pathetic in their ability to manifest a malignant state? It's likely the cells next to them suppress their malignant biology, and or they're waiting for that second signal to stimulate the malignant behavior. And so a fundamental question is, with the models, can we find the cell that cooperates with the cell that has the oncogenome to suppress its behavior? And is that different from a cell that actually stimulates its behavior? And so we study mesenchymal cells, mesoderm-derived cells, not blood lineage, but connective tissue lineage. The common term for these are fibroblasts. They're probably one of the most fascinating cell types in the body. They are frustrating to study because they're difficult to trace, and they may not display a hierarchy like the blood does and like other tissues in the body. They seem to respond to local cues to modify their behavior. There are these types of cells in the pancreas. One type is a stellate cell, and they differentiate into fibroblasts. And so we study how do these cells modify the behavior of an epithelial cell that carries an oncogenome. They make matrix, extracellular matrix, and we're pretty sure now the matrix influences the biology of the cell. Talks at this conference talked about touching by the developing embryo and how touching, modified the biology and the patterning, et cetera. It's same is true in cancer. The contexts, I think, of the cancer cell are going to turn out to be fascinating. So on the basic side, it's what's necessary besides the oncogenome to cause cancer. On the more applied side, it's can we use any of our observations to either detect that cancer early or treat the cancer, either as an early state or an advanced state. And the model systems allow us to ask that question. And as of now, part of my talk on Saturday is going to be about one of those observations where we have identified a pathway, because we have the models of health and every stage of disease, that allow us in a very, I think, systematic way to identify what's this critical thing that makes you the most malignant. And we have found a pathway which we think is going to turn out to be a relevant dependency in the cancer. We wouldn't have been able to find this pathway using methods that didn't, that we haven't, without the organoids or without the animal models. And so we're quite excited by that. So I do think that in the basic biology and the applied medical fields, these models have, really revolutionized things. And I'm hoping that the next wave of trainees can take them to the next level. And I'm pleased to see at this meeting we have lots of trainees.
Yeah. I mean, it's fascinating because now we're getting a broader picture of these cells. It's not, as you say, cell autonomous features alone. And I guess the question being is, when you're looking at these organoids and these tissue models, are you confident that they recapitulate what we're seeing in the patient? Because I think that is a key question some people.
So again, they only give me 20 minutes to give this talk on Saturday, so I can't actually speak about all of the things, but I would love to. We do have, when we first, when we had our first paper with Hans on the organoid models, we speculated that we could make an organoid from every patient and use them as a looking glass, as an avatar for their cancer, and identify a therapy that would work in the organoid, and then we would test it in the human. We're actually doing a randomized trial now using the organoids to do precisely that in pancreas cancer. And we have a clinical trial that's enrolled probably 2/3 of the patients, maybe a few more, in our randomized trial. And the trend that I see so far is that for the extremes, meaning organoids that are super sensitive to various drugs, patients look like they're responding to these drugs. And for the organoids that look they're super resistant to the drugs, patients don't usually do well when they're given those drugs. So the extremes look very promising for chemotherapy. Majority of the drug responses, though, are, I guess you'd call it the middle zone, where there's a mixed response. And this is where potentially, at least for chemotherapy, the organoids may not be so informative. Nonetheless, I mean, the extreme responders may be good enough to actually push the platform forward. And the point of this trial is to ask, are they better than a coin flip? And if they are better than a coin flip, in fact, if they look robust enough, then we can start to develop it as a laboratory-developed test, so that when the fancier therapies come along, the RAS inhibitors, et cetera, we will look for extreme sensitivities to small molecules and other types of therapies, and hopefully get to ask that question. So I think the question is still very possible, and Hans Clevers, who again I think deserves most of the credit for really putting the concept on the map. As you all know, he now has taken a post to run early cancer drug discovery and development at Roche in Switzerland, and they're starting an institute to do this. So when a major biopharma puts their resources behind, a really accomplished academic to take this big time, I think that says that, there's confidence that exists that this is not just a research tool. And so we're hoping that the little work that we can do in academia will stimulate this method forward. There's still a lot of work to do. I have to say, it's it's not the easiest type of experiment, but I am hopeful right now.
Yeah.
Yeah.
Do you envisage it being used clinically within as one of the toolkits before, in advising therapy or in this way, or is that too early?
Well, I mean, the best thing would be that you would like, any of these tissue models to be almost like a bacteriology test. So when a patient has a fever or has another infection, the doctor gets blood and sputum and other body samples in efforts to culture the pathogen and then do the Koch's postulate test on the pathogen to try to wipe it out. So they identify the bug and wipe out the bug at the same time, and then within a day or two tell the treating physician, it's this pathogen, please use these antibiotics. That's the general concept behind these models. Yes, can we devise an accurate pharmacodynamic method that will peer into the tumor to do these things? And it's formally possible. That would be the ultimate goal. Treat the patient eyes wide open, and we'll see. I mean, Cold Spring Harbor has invested massively in this concept. Many of the faculty here on our own campus now use organoids to ask fundamental questions and some applied questions like the ones we're talking about. I'm hopeful that we'll be able to push this forward. The organoids can be useful for studying developmental biology, aging, inflammation, cancer, maybe healthy tissue function too, like liver function, heart function, nerve function. We had a lecture yesterday by the scientist from Germany who generated the first brain organoids. Totally fascinating, right? And I think there's huge potential in this space, and for both basic science as well as applied applications. So I'm very optimistic still that this is a great area to work in.
That's very exciting, and I look forward to you reading more about and learning a bit more about the clinical trial results. Thank you very much, though, for your time.
My pleasure. Thank you all.